speaker, Professor Balvinder Singh. Let me take a chance to introduce him. And so thank you, sir, for accepting our offer for delivering a talk at Health Informatics Summit. So Professor Singh is a senior principal scientist at ACSIR, and he is basically heading the Bioinformatics Center at IMTech Chandigarh. So today he will be talking about one of the things which his lab is working for so many years now, where they are particularly interested in, the development of repertoire of small molecules against immune receptors and vaccine epitope predictions. So their lab is primarily using the structure-based approaches for this prediction analysis. So let me share with you a little bit about his academic journey so far. So Professor Singh has done his master's, and he's, in biophysics a long time back, and from, the Punjab University. And soon after that, he did his PhD in theoretical and molecular biophysics, again from the Department of Biophysics, Punjab University, Chandigarh. Now, he has been working in the Indian science for now probably so many years. So let me give you a very small glimpse about which are the premier institutes he has prior worked on. So he started as a junior scientist in the Bioinformatics Center in CCMB, Hyderabad in 1998. And later on, he moved to IMTech Chandigarh in 2002. Now, during this time, he was also involved, apart from research, he was also doing teaching courses like research methodology and bioinformatics for the pre-PhD ACSIR programs. Then he was also honored to work with a Nobel laureate at the Harvard University with Professor Martin Karplus. Not the least, currently there are 5 lab members, 5 graduate students in his lab, 1 postdoc and 1 research associate at the moment. And his lab is generously supported from the Department of Biotechnology, DST, ICMR, and many industrial research projects in the last few years. Now, let me also introduce you guys a little bit about his, what are the core things his lab has been working for so many years. So his lab has been using molecular modeling and simulations to understand one of the basic questions of biology, that is about, about the proteins with their interactions with their ligands and the protein and the DNA interaction. So his interest primarily lies into the protein sequence analysis, protein structure prediction, its annotations, its biomolecular modeling, and virtual screening. His lab also is particularly interested in one of the proteins where I am also interested, is the mechanism of transport of solute across membrane channels. And he's in that class, he's interested in aquaporins, which is widely known to be used as the drug targets. Last but not the least, his lab is also working on understanding the regulation and the substrate specificity about these channels for transporting the solute across the cells. So Professor Singh, I would like to request you to kindly share your science with us. And thank you very much once again for accepting our offer for sharing your science. Thanks a lot.
Thank you, Professor Gaurav, for giving the introduction for me. And thanks for the invitation from Dr. Raghav during this HIT Summit 2021. I'd like to tell you that we have been working in this area of molecular modeling, simulations, docking, and stuff like that for so many years. We have collaborated with many wet lab scientists as well. So to check whether our findings are online or not, or check their hypothesis, whether they are working, or to find out the mechanisms of various, protein interaction, protein small molecular interactions. During the last couple of years, we have also, worked on, in the area of immunology, whereby we worked on the immune receptors and the epitope prediction. We ventured into that area in collaboration with Professor Agrawala here. And this was a very recent, journey which we had in this area, and then I'd like to share with you. I have other stories also on the aquaporin and stuff like that, which we'll share on different platforms. As of now, we will, I'll focus on to how we have working in this area of immunology, immunoinformatics. Plus, Dr. Raghav also mentioned that we should also, this is a forum whereby we have to present what are the resources, what are the different things that we have developed over a period of time, so that the people resources or the methodologies or algorithm which can be shared across the community, so that they can be really used in the improving the health. I think that is the very good aim of this health summit, for that matter. So keeping that in mind, probably I'm going to restrict onto this immune informatics and stuff like that only. So Ecopony stories and other stories we'll talk some other time. So at this moment, there are two, kind of objectives on which I'm going to refer to during my talk. And these are the two lines on which we have worked during the last couple of years in collaboration with Professor Javed. One was basically we wanted to develop a small virtual library of the compounds, small compounds which can interact with the immune receptors. I mean, that library we have, created, and repertoire is there available, and we have made some kind of a contribution in this area. And also, we are able to, use the structural biology approaches for actually differentiating between the binders and non-binders, peptide binders and non-binders on the HLA. So these are two stories which I like to go through, so that I'll share this with you, how the molecular modeling or structural biology approaches can be basically used for such a thing. So, like coming back to the first thing, that is development of the repertoire. Hereby, I mean, what was the need of this? What was why we were creating the repertoire? So many small molecular libraries are available. So we wanted to create a specialized library and wanted to see that does it make really sense while analyzing this library. One, we created the library. Second, we tried to analyze what are the different compounds that are there within this library, and how these do they really make any sense to us or not. So this is what we attempted when we made this library. I mean, I think most of us are aware of that immune receptors are very important. They are involved in a lot of disease, abnormalities, morbid conditions, autoimmunity, cancer, and developmental disorders, infectious diseases. I mean, I have not written all of them. So, but you can imagine the asthma, allergies, all these kinds, they are basically a part of association with the immune receptors. Available therapeutic interventions, which are there, they are adjuvant therapies, antibodies, synthetic ligand-based treatments. So they have demerits. That's why we have an opportunity to create some kind of a specialized, carry out a specialized task to find out the small molecules, so that these molecules could be used by the experimental people. We have done purely a in silico kind of a study here, and these small molecules, they have been proven that they interact with the protein molecules. They can change their activity. They can change their whether they are morbid conditions or otherwise, they can influence the activity of any protein. Most of the immune receptors are protein in nature, so we wanted to affect their biological activity, particularly in the when they are in the morbid conditions. And there is a kind of a urgent need to do that, and we wanted to go ahead with this. And traditional ways of finding this is you take a chemical library, you synthesize the molecules, or you get the synthesized molecules and try to experimentally by using hit and trial kind of experimental methods, and try to do this kind of a check and recheck with the help of a medicinal chemist whether these compounds work or not. So this becomes very expensive, laborious. And these days, in silico techniques are at hand, and you can do wonders with that. Computers, their power of the computer is unlimited as of today. I mean, libraries are available which offer different kinds and sorts, and these chemical virtual libraries are available. That's what I mean. These can help you to play wonders with these with the with particular any therapeutic molecule, for designing any therapeutic molecule for looking at the interactions. So a lot of benefits are there for the structure-based virtual drug design. So virtual screening, basically drug design is a separate part. So at this moment we were looking at the virtual screening, reduces time and cost, and large amount of chemicals you can just quickly you can go through the screen them across the different protein molecules. Libraries, you do not have to have access to the physical library. You just need a virtual library and you can do this thing. You only require some basically a manpower and computer resource, and you will be able to get the desired or most likely of the potential compounds. You can hit those and then you can go for the real chemistry and things like that. So in the present study, we have taken about 25 immune receptors. I mean, we have created a repertoire of about 5,000 small molecules that exhibits efficacious binding and distinctive drug likeliness. We'll talk about this. So we have selected high resolution structures of the immune receptors. So these structures are mostly available in the PDB. Idea was to take 3.5 Armstrong resolution of a structure, if that is available with us in the PDB, whether it's in a complex form, co-complex form with the peptide or not. So we got them for the innate receptors, major histocompatibility complexes, co-stimulatory molecules, co-inhibitory molecules, cytokines, addition molecules. So all of this information was basically obtained from the PDB. Wherever we could not get the structure from the PDB, that is crystal structure, we used homology models. So wherever we could get a good homology model with a good strength and good confidence, we use those models to carry out our further work. So basically, we needed a structure because we wanted to go for a structure-based virtual screening kind of approach. So we looked at them. We looked at these structures carefully. As you can see, we have clubbed them in different categories as per the classical way of looking at the immune molecules. And then we performed the virtual screening. Virtual screening is easy. You have to perform just docking, but before that, you have to do many steps. Like you have to see whether the molecules are good enough, in a good shape, ligands. So prepare the ligands. You prepare the receptor molecules, and then we look at what are the binding pockets. So in the previous slide, I've shown you that the regions which are blue colored here, they have been indicating of the ligand binding site. So here I'm talking about how we did that. I mean, in a sense that we looked at if already co-crystal structure is there, we looked at those crystal structures, and then we try to get the information about the amino acids residues which are interacting. That is 4 Armstrong to get criteria, and within that we try to see that how many residues are there, and we use them as a kind of a reference pocket for interacting with the ligand or the peptide. For others, whereby we didn't have this information, we use the in silico prediction servers, and these prediction servers helped us to predict those binding pockets. So once we have this information of binding pockets with us, we have the information of receptor, we have the ligands with us. We, took the zinc library here, and we went for the scanning. We just, scanning in the sense that we perform the molecular docking. So molecular docking you perform, you get a docking score. So docking score here is in the form of free energy of binding, delta G. We found out for all the molecules. We can see that how much of is it really a interaction which is there is. Negative value basically shows that the average free energy of the binding is good enough. But how much more negative it could be, that more the negative, more it will be better. If positive value is there, it doesn't have any meaning for that. So we were able to look at the average free energies of binding with the standard deviation for all the 200 molecules against the respective immune receptors. For each immune receptor, now we restricted to about 200 small molecules. We could have gone for more also, but we found that 200 is good enough to take them, because as we did the analysis further, you realize that this was a good number to begin with, to have some kind of a screening for that matter. Then we went for the clustering of these molecules. Wanted to see that how these molecules are with respect to each other. So we went for multi-dimensional scaling and the clustering on the entire group of small molecules for a particular immune receptors. So we performed this analysis. We obtained this in some kind of a 3D plot, whereby each we bin them based on some kind of structural diversity among these small molecules. Depending upon how much is the difference between the two ligands, two small molecule, which can interact together, which are structurally together, they are put in one bin. So we distributed them in basically different bins. Each bin has a different variables, and all these bins. And on this, you can see that this is showing the multi-scale dimension, multi-dimensional scaling. So just structural samples. So wherever the dots are near to each other, it means they are mostly forming a one group. Whereby they are away from each other, so they are different from the rest of them. So like this, it will be easy for us to segregate based on some kind of coefficient, and you can just segregate them in different bins, and then for each bin you can see that what are the different molecules which are present. So then we can have a representative of each of the molecules. Representative can be based on some kind of a tonometer coefficient we can take, similarity coefficient. With a cutoff of 0.4 or on the small molecule, we can see that what are the different representative structures we can get from different bin. So largest bin of the individual immune receptors, you can find out what are the representatives. That may not be best. So we are providing you the next total 400 compounds for each one of the immune receptors, so that you can just go through the whole of the library and select the particular compound. If you want to go for a really variety of the compounds that for a particular immune receptor. Also, we looked at the ADME properties, which reveal the basically drug likeliness of the small molecule binding to the immune receptors. So ADME enabled us to predict major physicochemical properties, lipophilicity, water solubility, pharmacokinetics, and drug likeliness, medicinal chemistry. All these properties have been considered, and they have been put in the basically in a Venn diagram here. So wherever you see the central number in these figures, you can see these figures. The central shows that they have the kind of all the common properties are there within those among all the three different set of filters which it went through. Different set of filters. I think everybody knows that we can talk about Lipinski, Ghosh, Weber, again, and Muget drug likeliness filters. And these filters, if you plot, then you'll be able to on the Venn diagram, you'll be able to see what are the molecules which are clearing all the filters from here. So it will be easy way to depict how many are there and which one are there. So all this information is available in this resource, which we called as Immutor Lig DB. So it provides access to the ADME properties of each ligand. So you have ability to choose that which ligand you basically want to go for. And this is the kind of overall architecture of this. And those who are interested, who are students and they want to build the databases, MySQL server and HTTP server, they have been used. The front end is that, the back end, basically MySQL contains all of this information. So firstly, we make the PDB search, and we do the homology modeling wherever required. And we get otherwise we get the information from the crystal structure, go for a pocket analysis, and then go for the structure based virtual screening using the Zinc database. So you use Zinc database, we use the drug-like compounds from this database, and then we rank them and catalog them against each of the receptors. And you can view these things on the ImmutorLink DB. 200 molecules, small molecules have been saved for each of the immune receptors. Additionally, we wanted to check that whether what we have screened, are these molecules good enough, how good they could be. Are they really different from each other? So we wanted to check the novelty. And there was also a question that there are already some of the compounds are existing. In PubChem database, some of these compounds are already existing, which are experimentally tested. So if can we compare whatever we have, predicted, or these 200 molecules for each of the, immune receptors, we try to make a comparison between them, basically. And this comparison is represented through this heat map. It shows that wherever this nanometer coefficient scale is kind of a 1, this is basically a good match. Wherever it is, green, it is it's not a match. So most of the heat map you see, basically it is green or a yellow. So that means basically we are able to hit some kind of a novel molecules here. So that is the purpose of looking at that comparison with experimentally testing molecules. So are we finding something new here, or just we are creating a library which may not be of that use later on? Also, we wanted to look at that, these compounds, relatively, how do they, behave in terms of their structures, where they are. I mean, if they are compared with themselves or something like that within the each, uh, immune receptors, what we have screened, are they really diverse? We find that majority of them, they are diverse. Only 0.02% exhibited high structural similarity, that is threshold of 0.5 to 1. Otherwise, these are kind of a very unique molecule. They have been catalogued in the hematologic, DB for this purpose, and they are structurally different from the experimentally tested ligands. This is what we are able to analyze performing this library. Now, this repertoire is freely available on this, on the website. That is bioinfo.intel.res.in BBSM. Easily searchable. I think graphical user interface makes the things very easy these days. So you can just search through the compounds. You can see what are the compounds there. You can draw and design the compounds, also play with them on this repertoire. So this is a kind of a summary, quickly going through. 5,000 total compounds are there for against the 25 important immune receptors. Free energies are negative, so confidence is there that free energy is negative. They are away. They are not similar to the experimentally tested compounds so far, whatever is available in the PubChem. And these are not having kind of similarity among themselves. So this resource can be a good learning platform for doing any computational developing any computational models for the small molecules in the immune targets, or even for somebody interested to use this for further development of the drugs, of the molecules, of the design. This can be, they can serve as a good scaffold or the primary structures whereby these can be used for further interactions and further drug designing effectively. I think this is the first part which I wanted to talk about. The second part was that basically that we developed a kind of a pipeline to differentiate between the kind of binders and the non-binders of HLA class 2. So there are a lot of information is already available in this area. I mean, what we found that how we can be different, can our approach be different? Can we look at this and look at it from different angles? So we had something in our mind. So our lab is a structural lab, and we talked with the immunologists over here, and they were very much interested that there are already so many sequence-based methods are available. So can we have a structure-based method which can be basically used to predict the epitopes? So I think I don't have to go much into the vaccines and stuff like that, why they are important, why do we looking for epitope? Because during this COVID time, a current pandemic, a lot of talk has already been going on about that. I think everybody is aware about vaccines because everybody is has got vaccinated by now. And most of us who will be attending this talk, I think they know what these vaccines are there, how they are being developed. But there are classical ways, like attenuated vaccines are there. There are epitope-based vaccines are there. I mean, these are the things that are there, and people have been quite, familiar with this. But how in silico, techniques or in silico methods can really help in making the progress in this area. A lot of servers, Dr. Raghav is there, and his group and a lot of other groups internationally have developed a lot of servers which are, whereby they try to, they exploited the sequence features to predict the epitopes. They exploited the, tried to exploit the structural features also. So whereby trying to make some kind of a mark so that we find out a epitope, right epitope to make a vaccine. So every type of algorithm, they have their own, pluses and minuses for that matter. Of course, good thing is that they can give you quick answers. Whether you get more false positive, false negatives, those are the matters of basically discussion. We can discuss these methods are good or not good. But I mean, we wanted to look at this. This problem from a different angle. We had some idea, and we thought that we will try to see if this angle works, and we can make a mark in this area. So basically, we wanted to exploit the structure. We were saying that if you use different servers, you get different kind of results. And actually, if you go to the bench, many of the immunologists will disagree with the in silico experts for that matter. So their prediction servers good, or they're bad. Sometimes works, sometimes doesn't work. So, but there is always some kind of development is taking place, some kind of, that's how the learning probably takes place, and then we move forward in the science. So we wanted to move to use the structure-based approaches to look at some of the epitopes, and can we really make a mark on that? So what we did is we looked at all the structures. We took 30 co-crystal structures of HLA binding from PDB. Our target was basically HLA2, that is class 2 HLA. There we wanted to take only those binders, only those structures where binders are of size not less than 10, because you require some kind of a bigger length to bind to the particularly this HLA type. So we looked at looked through the PDB. We found that there are 30 co-crystal structures are there, and with the binders having size not less than 10. With these conditions, 30 co-crystals we had. 20 were presented on HLA type DR, 4 were on HLA type 2, HLA2 type DP, and 6 were on type DQ. So by looking at this, we wanted to see that how do these peptides, we had to have the structures of the peptides. So we were interested, is there any common in the structure of these peptides? So we tried to look at the structure. We tried to look at the phi psi, particularly at the atomic level, that what part phi psi these amino acid residues of the, we divided them in different mers, 22 mer, 14 mer, and 13 mer, 20 mer, 17 mer, 11 mer, or something like that. So depending upon how many we are able to find, how many structures we are able to find from the PDB, we try to analyze their phi and psi, the diagonal angles. Phi and psi are through diagonal angles of the amino acids. We try to look at those. We try to see that what kind of diagonal angle do they adopt. Is there some kind of a, some kind of a commonality or similarity that we can look for? So we try to, there are a lot of variabilities. Within that, we try to find some kind of a high second order polynomial function to find out some kind of structural latentness. More or less, we are able to find, if we look at the, particularly this polynomial, except for the 11 mer peptide. So the 11 mer peptide was the kind of exception. Then parallelly, we were also interested to look at the B factor analysis of the for the amino acids of the peptides. So B factor is the one which is given in the PDB, given basically reduced by the crystallographers. And the PDB, they mentioned that what is the B factor for each and every atom, or particularly for amino acid you can compute. It tells essentially basically the mobility, dynamic mobility of particular amino acid residue. So according to the B factors, we try to color them. Color is basically if the low factor, if the B factor is lower, it will be blue in color. It will be, if it is high B factor is there, and it will be red in color. What we did is we couldn't have checked the B factors across all the peptides. So within the each mer, that is 22 mer, 14 mer, we normalize the B factor. We normalize the B factor, then we try to see that can we make some kind of a groups? Can we see that what kind of regions which are there with having some kind of a consensus we can reach at. So looking at the size, we try to look at that how the B factor is basically varying for each the amino acids. So more or less B factor correlates with whatever the structural or the phi and psi deviations are for that matter. Basically, if you try to see the central part, the core region, the B factor lowers over there. The striking regions are having high B factors. That is quite kind of a normal, but this is what kind of expected also. So, in addition to looking at these peptides, we thought that why don't we look at the different types? If there is some variability in the peptide we are looking, that's fine according to their sizes. Should not we look at the type of HLA, DRB1, or their subtypes? That if we take those and the peptides and try to see that what kind of a structure, what kind of any other information that we can get from this. So 9-mer region, what we are able to find out is the kind of a one which has been basically highlighted in yellow here, and shows that basically we found that this is the region which is more or less remains the kind of the same, except the flexible flanking regions. But highly flexible flanking regions have been found in some of the peptides you can see visualized from here, PEP8, PEP12, PEP13. So these are some of the nitty-gritties that we also went into these peptides when we looked at the structures. What we found is the mean value of the psi is basically of the subtype. The exhibited similarities grows across the diagram, except for the position 3. So psi scores were more or less kind of a common, and phi also share the same values except for few of the positions. Some variability is there in the phi angle, some not much of a variability is there in the psi angle. So these are the kind of observation that we obtained from this this study. So this is just we are trying to make sure that what we are doing, looking at the core region, is basically what information take-home messages are there. 9-mer core is a kind of a good central part structure. It's basically a stable structure more or less as compared to the flanking region. So wanted to see that, can we look at the type of amino acids? If you look at the type of amino acid, is there any kind of a similarity we can make out? Perhaps none. If you plot go for a logo plot and you'll find the frequency for each position at the 9-mer. There's not much of a commonality. A lot of variability is there within the amino acids. But if you, you have to look at from different angles. So what we thought is, why don't we look at we plotted in fact this information for the different subtypes also. There we found some of the similarities, but not very stark similarities were there. So logo plot was also constructed not on the amino acids, but according to the subtypes also, with for the peptides for different subtypes. And also, we looked at these from a different angle. We looked at polarities, non-polarities. So phi and psi, we looked at these angles for polar uncharged residues, negatively charged residues, positively charged residues, aromatic residues. So we wanted to see that we should be able to find out some kind of a commonality in somewhere, so that can be used some kind of as a kind of a hint, or some kind of a leverage point, anchor point, from where we can just grow and build up a structural some kind of a method, so that that can be used for building up this information. So all this analysis showed that predominantly it is going to be a linear HLA peptide, devoid of any particular kind of a structure. For that matter, this is what we were looking at. There were 9-mer region at the core in the flanking region. Not very great information, or it's very clear-cut information is there in terms of amino acids, the type of amino acids that could be there at the different positions across these available structures. Then what we did is we wanted to look at basically the aim was ultimately to do docking. So in immunology, basically they say that this CLIP peptide, that is HLA invariant chain peptide is there, which had to be replaced by the antigenic peptide. And this peptide exchange mechanism occurs within the compartment of lysoendosomes. And this we want to check that are the antigenic peptides that we have are able to do that. In a sense that can we find out if we do the docking and compare the delta G CLIP versus the co-crystallized antigenic peptide? Are they getting the more stronger, showing the more stronger affinity? This way we compared and we found that for 26 of the 30 available structures that antigenic peptide is stronger, showing the stronger affinity in terms of negative delta G as compared to CLIP peptide. We also did RMSD checks across the peptides. These peptides have been redocked. Peptides have been taken and they have been redocked. What we found is RMSD is quite kind of a good enough. So it means kind of a docking can work. We also looked at, we compared the redocking of the CLIP also. So CLIP peptide was also redocked and we found kind of a similar variability CLIP versus the antigenic peptides. Low range of RMSD has been found out in the molecular docking for the antigenic peptide as compared to the CLIP peptide. Because maybe the data is low over here for CLIP peptides, not much of the data is there. For antigenic peptide, we have a lot of data. So, but the take-home message was probably we can use the molecular docking and for testing, for checking with reference to CLIP whether our antigenic peptide could be good binder or not, with more efficiency it can bind or not. So, as we know that basically in the lysoendosome compartment, there's a acidic pH. So definitely we know that the conformation peptides are very notorious. If you do the molecular simulation, then try to see, even if you do the experiment, if you talk to the peptide chemist and otherwise peptide persons who work with the peptides, they see that there's a lot of conformational changes there if they change the environment of the peptide. So they are likely to change a lot of, conformation. The type of conformation, same peptide can be helical, same peptide can be a beta sheet or otherwise, and if you change the solvent. So what we found is that most of these servers, whereby we are predicting the peptides, the initial servers, they like PEP4, they work at kind of a normal pH 7. Though they never take care of pH 4.5 or 5, which is the actual pH at which basically this peptide exchange mechanisms happen. So keeping this thought in mind, so we thought, can we use MD simulations here? And using the MD simulation we carried out on pH-based MD simulations we carried out, and we tried to see that how can we achieve a structure which is close to the crystal structure, which will be a linear structure if we use a pH of 4.5, because if we use the neutral pH or other pH, the structure was not very close to this thing. And we began with a kind of a linear structure of the peptide, and we tried to do the simulations and see that the neutral pH, there were conformational changes happening, which were not making it away from the from the crystal structure, which is co-crystallized peptide structure. But with pH, if we keep 4.5, then we are able to see that they are very near. They are not, of course, very near, but they were quite comparatively, they were near as compared to pH 7. So MD simulations worked fine at pH 4.5, and we were able to compare their RMSD, compare their radius of gyration, and looked at made the comparison with the crystal structure peptides. And in 26 out of 30 peptides, cluster representative. So MD gives you large number of structures. If you do MD simulations, it gives you large number of structures. So you have to choose the structures. So what we did is, again, we did some kind of a clustering on that. So we took the different cluster representatives. And what we found is that out of 30 peptides, for 26, the cluster representative had an RMSD of crystal structure which is less than 5 Armstrong or so. So which was quite kind of a good enough if we look at compare the structures that matter. And we attempted to use this information for further analysis and work. So what we did next is we took test peptides from the PRIDE database. And these test peptides are known test peptides, and they known peptides which with established immunogenicity. We took these peptides. They were basically binders, but we took also some non-binders, which are typically neoantigens, and they were not basically known binders for that matter. And then we went through the same procedure, and we did for docking, did for went for pep4 analysis, went for pH-based MD simulations, and try to see that where the structures are same. So more or less, when we did this comparison, what we found is, and with some kind of interpolation and looking at the affinity scores, we are able to find that across the three allelomorphs, we were able to say that the straddle angles of the peptides, they indicate dissimilar conformation for the non-binders in comparison to the crystal peptides. So this is for dissimilarity for the non-binders, we were able to distinguish quickly, quite all right. And we try to substantiate this information in terms of low efficacy of non-binding test with the peptides as compared to HLA binding test peptides. So this test was also done by carrying of the docking. First, we did the comparison of the lateral angles, then we did the docking and try to find across the three allelomorphs, and this results are better. Moving forward in this area, we found that out of the 9 instances, that is approximately, you can say 77% of the time, non-peptide test binders, they had better efficacy to bind to HLA allelomorphs over the non-binders. Over the 2 instances, where the non-peptide binders had better affinity, efficacy, and reasons for this can be looked at the lateral angle, some variability, or you can say the less number of structures that we had with us. That way we could not, get the 100% results over here. In addition, when we submitted this paper, referee also asked us to look at the, peptides or the immunodominant epitopes of the SARS-CoV-2, because that was the kind of a beginning of COVID in other countries at that time. So we looked at we took these epitopes also, we tried to, went through the same protocol of pH-based MD simulation, structural analysis and docking and stuff like that. And we found that the affinity score is kind of a -20. I mean, out of the kind of a clusters of the peptides. So in a sense, there may be certain difference in the orientation of the HLA peptide, but the use of the pH-mediated MD simulation coupled to the docking can be a potential method. That's what we were kind of arriving at. So in those lines, we have given the schema whereby one can take a sequence and generate overlapping peptide fragments potentially, and then we can, if we can subject it to some kind of pH-based molecular dynamic simulations, make the clusters, choose the clusters, and then put them to different filters of statistical analysis, looking at the psi angles, looking at the different filters that analysis that we have gone through. And then finally, we can carry out the docking and sort it with reference to CLIP. We can say MD simulated peptide versus CLIP, whether they are good binders or not. This way, a new basically algorithm can be used for making predictions for the epitopes, which will be purely kind of a structure-based kind of things. Of course, there can be arguments that this method works. Actually, we wanted to make this kind of a server online, but we couldn't do that because the student has to rush and then things like that. The COVID, it was beginning of the COVID, so many things were happening that time. So we stopped here, and only I'm presenting the way that we follow. These are the contributors that were there for the IMTOLIC DB. The Kayan Chatterjee majorly worked. Gurkirat Shilpa also helped a lot with this. Dipayan was also a contributor in the epitope prediction work. There is structural analysis. Pragya helped us in carrying out this pH-based MD simulations. And we had plans to, carry out some kind of experimental work also along these lines to check whether what we are predicting is right or not. But somehow that couldn't work. We did the experiments, but somehow, I mean, we couldn't, get the desired results, you can say, for that matter. Khurram was there with us, but I mean, this was a paper, was a kind of a dedication to him. I mean, then Javed's help and interaction was also very much contributing to this work that we had carried out during previous years. I think I'm done with that.
So thank you, Professor Singh, for your stellar talk. So I think now we can have